Hello, my name is Kevin Eberhardt, and today I'm going to be presenting my group's gut microbiome study. I'm going to be talking quickly because I want to fit everything in. So, before I talk about that, let's talk about what a microbiome is. A microbiome is the collection of microorganisms and overall genetic material that reside in a given location. This includes fungi, bacteria, archaea, and single-cell eukaryotes. The gut microbiome consists of tens of trillions of microorganisms that live in the intestinal tract. So that's the gut microbiome, which we'll be talking about today. Its function includes helping the body digest certain foods, regulate the immune system, and protect us from pathogenic invasions. Every individual has a unique gut microbiome, and this plays into our physiology. Studies have shown correlations between the impacts of the gut microbiome in human metabolism, nutrition, as well as diseases. Having an imbalance of microbes can lead to gastrointestinal health problems, such as type 2 diabetes, obesity, and inflammatory bowel disease. Although type 2 diabetes can be inherited, you are at risk of getting it if the composition of the microbiota is not like the average person, like having a decline in the butyrate, producing bacteria that regulates energy metabolism. Over generations, our microbiome has adapted to environmental and diet changes that also affect our mental health. The state of our microbiome not only affects our immune system, but also the gut-brain connection. The inflammatory species that are a product of species of bacteria interacting with the endocrine and immune system have direct communications with the brain due to self-produced neurotransmitters and nerve receptors in the gut. Maintaining optimal levels of vitamins and minerals and healthier diets is key for better brain functioning. So the gut microbiome has a direct effect on brain health. There's a gut-brain connection there. In our study, we're asking the question, how does the weight of German Shepherds affect the gut microbiome? Or how does the gut microbiome of German Shepherds affect their weight? We hypothesize that heavier German Shepherds will have higher amounts of bacteria that have been associated with obesity, such as Firmicutes. So, we have 4 German Shepherds in our study. They are all healthy German Shepherds. They all exercise regularly and have comparable diets. Sample 365 is an 80-pound, 10-month-old male. Sample 406 is a 70-pound, 1-year and 5-month-old female. And sample 521 is a 67-pound, 1-year, 4-month-old male. And sample 643 is an 80-pound, 2-year and 5-month-old male. So those are the differences. We tried to get 2 that were in the 80-pound range and 2 that were in the 70-pound range so that we can compare the differences between them. Moving on to materials and methods. First, in our procedure, we isolated and concentrated the DNA. Isolated the DNA using a QIAGEN kit, and then we concentrated the DNA tenfold using a Zymo kit. The isolation homogenized the stool sample in a buffer that prevents enzymes from degrading the DNA, binds the DNA to a filter, washes out all of the impurities essentially, and then eventually elutes the DNA from the filter so that we have what's hopefully just DNA. Then we concentrate that DNA tenfold using the Zymo kit and rinse it of impurities in a special buffer. So after that, we use UV spectrophotometry to make sure that we have a good concentration. We use 260 nanometer and 280 nanometer light. We actually use a nano drop to do this, and that ratio, if it's in the 1.5 to 2.2 range, means that it's likely pure DNA that we have gotten from our sample. If it's below the 1.5 range, then we probably have some protein contamination. Then we move on to PCR amplification. So we isolate the DNA already, then we have to amplify it. Specifically, we're amplifying the 16S rRNA gene. Now, this gene has variable regions that are interspersed between conserved regions. These variable regions are frequently used in phylogenetic classifications such as genus. So the variable regions help to show us what are the different species or what are the different genus, in this case, of bacteria that are within the gut microbiome that are being displayed by the DNA that we've collected. PCR amplification involves 30 to 40 cycles of amplification, and then these are put in a thermocycler. So basically first it's denatured by heating up to 95 degrees. Then the primers bind and the temperature is raised to 72 degrees. Heated up to 95 degrees, cooled, and then raised to 72 degrees so that the DNA polymerase can bind and start synthesizing. So this is done 30 to 40 times so we get a large quantity. We then use, in the positive control, we use E. coli, and in the negative control, we add no DNA. We then do gel electrophoresis to determine if this worked. Gel electrophoresis, as you can see over here, you have a positive end and a negative end. Current flows through. You put the DNA in, or what you hope is DNA, on one side, and then the molecules will be pushed through by the electric current. So if you get these bands here, that means that you have successfully concentrated or amplified your DNA. If you don't get the bands, it means that you have not. Then you engage in amplicon cleanup, PCR cleanup, which removes all of the impurities other than DNA, and then we index the PCR, which is very important. This means that we add a sequence of tags to the end of the amplicon from each sample so that it can be indexed, and then that allows it to be sequenced later on using a fluorometer. So fluorometric quantification and dilution is the last step here. And we use a fluorometer because UV spectrophotometry would not work for a sample that is that small. So moving on to our specific results here. In terms of our initial, after isolating and concentrating the DNA, we determined that our DNA was okay. This is actually not our results specifically. This is from another group, but we followed the same process and had similar results. All of our ratios fell within the 1.5 to 2.2 range, so it was safe to assume that we had relatively pure DNA, which is good. We then used some PCR calculations that we made in order to create solutions of 50 microliters. We used Maxermix, extracted DNA, and we used the concentration of DNA to determine how much DNA template we would put into the solution, and then we put water as the remaining to get the volume up to 50 microliters. So, as we can see here, once we engaged in the, so then we did gel electrophoresis. As you can see with gel electrophoresis, our negative control does not have a band. That's what we're looking for because our negative control is just water, it doesn't have any DNA in it. This also means that there was likely no cross-contamination during the addition of the DNA to the gel, which is good. Our positive control and samples 643, 406, and 1048 all had bands, which means that the amplification of DNA was a success. However, 365 did not, so that one didn't work, unfortunately. Moving on to our results here from the actual sequencing. All of our samples that we used for this section, our actual samples due to time limits within the class, did not go through, but the samples that we used had over 10,000 reads, all of which passed quality filtering. Then if you can look at our kingdom level analysis here, all of these samples are 99.6 to 99.8% bacteria, which is really good. That's exactly what we wanted. In terms of the positive control, moving down to the order level, you can see that it's almost entirely enterobacterialase, which it should be because that is the order level of the E. coli bacteria. And then over here at the order level, bacteroidales and clostridials were found in high levels in all samples except the positive control, as I mentioned. There was, however, no recognizable differentiation in the quantities found that correlated with the weight of the samples. Moving down to the genus level, at the genus level, Prevotella was the most common strain in all samples except for dog 3, that's sample 521. In dog 3, Bacteroides was the most common strain, and that was the second most common strain in all other samples except for dog 2, which had a high concentration of fusobacterium. Once again, weight did not seem to correlate to concentrations of bacteria within the samples. So, in our discussion here, a lot of this I have already mentioned. We had proper ratio 1.5 to 2.2. Our gel electrophoresis indicated that we successfully amplified except for 365 and for the negative control because they did not have bands, although there was likely no cross-contamination. Each of the samples that we used in sequencing had over 10,000 reads, which is why we selected them for analysis. However, if you look at the genus level of the positive control, we're only at 82.14% Escherichia, which means that there could have been some cross-contamination there, because that level should be higher, because we're looking for pretty much entirely Escherichia, because it should be mostly E. coli there. When you're looking in here, we have some of them listed as essentially unclassified or unclassified at the order level. So these are bacteria that would not fall within the classifications that we had before. In terms of our hypothesis, the results could not prove or disprove our hypothesis. There were no notable similarities. Basically, weight did not seem to correlate to any of the differences in bacteria that we found. As I discussed, we did have some notable differences and some common themes like clostridiols and bacteroideals. I butchered that one. But the different bacteroids, which was present in all samples, has been associated with normal weight in human children, according to a study that I found, which is interesting because that might mean that our German Shepherds were all relatively normal weight, and the reason that we weren't seeing differences is that we were hoping to see was because they were relatively normal weight and not spread out enough, and not large enough across the spectrum of weight in order to really see differences. We're only talking about a 10-pound difference, so these were likely normal, healthy dogs, which is why we didn't see the differences we were looking for. Thanks for watching my presentation. Hope it wasn't too long, and have a great day.